Good morning, everybody, and welcome. So today I'm going to rush through HIV. To start with, I thought I should probably just define HIV to make sure we're all on the same page. So HIV stands for the human immunodeficiency virus, and it causes acquired immunodeficiency syndrome, or AIDS. So HIV is the virus and AIDS is the disease. And so I'm going to tell you a little bit about the discovery and origin of HIV. I'm going to tell you a bit about the basic biology of the virus, which is what I work on. I'm going to talk about the treatment for HIV, which I think is really one of the major success stories of modern medicine. And I'm going to tell you what still needs to be done. So the reason that you've probably all heard of HIV is because it caused a pandemic. So a pandemic is when an infectious disease goes around the world, usually resulting in many deaths. So this figure just shows you the estimated number of deaths from different infectious diseases in history. And you can see that top of the list there is the Black Death, caused by a bacterium, which we now have a vaccine against, so that isn't a problem anymore. You can also see smallpox on here, which has been eradicated. But coming up at sort of number 5 in this list is HIV and AIDS. So in total, HIV has infected 84 million people. And it's actually killed nearly half of those people. In fact, left untreated, HIV would kill almost everybody that was infected. And today we have about 39 million people living with HIV, and we've got that number of deaths down to less than 1 million a year. But probably one of the most worrying numbers on that slide is that there are still millions of new infections. So in the latest data that we have in 2022, there were 1.3 million new infections of HIV. And you can do the maths there and see that actually the number of people living with HIV is actually increasing year on year. So when did we first become aware of HIV and AIDS? Well, that was over 40 years ago now, back in 1981. So in those days, there was no social media. I think there wasn't even the World Wide Web. So the way the news was spread was through something called the Morbidity and Mortality Weekly Report, which was published by the Center for Disease Control, the CDC in America. And in the summer of 1981, they reported some very strange cases. Young, otherwise healthy men were coming into hospital and dying of pneumonia. This normally only affects the very young, the very old. And so this was very, very strange cases. Not only that, but many of these people also had a very rare cancer called Kaposi sarcoma. And this was a cancer that was mainly found in old men in Africa. So this was sort of strange, and so they reported it as strange cases. But the number of cases started stacking up. So mainly these were young, otherwise healthy, often gay men. And they were coming into hospital with what we call opportunistic infections. So these are infections that normally wouldn't cause a healthy person any serious problems, so things like thrush. But in either the very young or the very elderly, or importantly, in people with a weakened immune system, these then can actually be fatal. And so it wasn't until about 1982 that this was given the official name of AIDS, because scientists had then looked at the epidemiology, so how this disease was kind of spreading, and they realized that it was probably caused by an unidentified infectious agent. And finally, in 1983, so 2 years after the first reports of this new disease, a group in the Institut Pasteur in Paris isolated a virus from a patient with AIDS. And they initially called this virus LAV, which was lymphadenopathy-associated virus. And a sort of little while later, another group at the National Cancer Institute in America, led by Bob Gallo, also identified a virus, and they called this HTLV3. Some years later, these were found to be the same virus, and they were renamed HIV. And actually, François Sanusi and Luc Montagnier here received the Nobel Prize for this discovery 25 years later. And then a couple of years later, in 1985, people realized that this virus was actually linked to other diseases as well, in particular slim disease in Africa. This is a sort of wasting disease. And by this time, we'd realized that it wasn't just gay men that were succumbing to this disease, but hemophiliacs, people that were intravenous drug users. And in fact, we realized that actually anybody could catch this disease. And I think it's important to say that actually at any time in history, there's been more heterosexual people infected with HIV, and particularly in Africa. And by 1988, actually, HIV and AIDS had reached epidemic levels in many African countries. But the African leaders were quite hesitant to admit this or even talk about it. In fact, mentioning HIV was banned in many countries, and you weren't allowed to put AIDS down on a death certificate. This was mainly because they didn't want to induce panic, but also they were worried about damaging tourism in these countries. But the reason that the world was sort of slow to appreciate HIV and AIDS is really because there's three main phases of the disease. So this graph shows you virus levels in blood in the red line, and then CD4 T cells, which are white blood cells, part of your immune system, in the blue line. And this is the sort of stages of infection. So initially, we have a spike in the amount of virus, a peak of virus in your blood. And this gives normal viral symptoms. So actually things like rash, fever, vomiting, diarrhea. And you have this as an acute infection for a couple of weeks. But then you have this long chronic phase of the disease where actually there are no symptoms, and most people then don't realize that they're infected with HIV. But during this time, your CD4 T cells, these white blood cell levels, are decreasing. And eventually, they reach such low levels that you start to succumb to these opportunistic infections, and your immune system is so weakened that you can't mount a defense against these agents. So really, it's this depletion of the CD4 T cells that leads to immunodeficiency and ultimately death. But during the chronic phase of infection, actually, an infected person can actually make 10 billion new viral particles every day. And this actually gives the virus multiple opportunities to modulate, to mutate, and to adapt and evolve. And this is one of the reasons that makes HIV so difficult to deal with. So when HIV was first discovered, the most closely related virus to HIV was actually found in sheep. It's called Visna. But soon after that, many similar viruses were actually discovered in a whole range of different monkeys. So these viruses were named Simian immunodeficiency virus, SIVs. And actually, there's at least 40 different monkey species have been found to have their own version of HIV, so their own SIV. And by doing lots of sort of sequencing of viral genomes and comparing the HIV genomes that we have to monkey viruses, we found that actually there are two kinds of HIV. There's HIV-1 and there's HIV-2. And actually these are related, most closely related to different SIVs from different monkeys and indeed apes. So the origin of HIV-2 seems to be sooty mangabees, shown here on the right, whereas the origin of HIV-1, so HIV-1 is most closely related to an SIV that infects chimpanzees. So this was somewhat puzzling, and people were wondering how we could catch viruses from monkeys. And I know at the time there were many theories, sex with monkeys. I'm sure people came up with their own theories. But probably the most likely explanation is the African bushmeat trade. So in many countries in Africa, monkeys are actually hunted and butchered for meat. And these rather gruesome pictures here show you that these processes involve large knives and lots of blood. And so one of the things that I didn't mention about the monkeys is that actually they have very high levels of virus circulating in their blood. But interestingly, they don't generally get sick, so they don't really get AIDS. But because they have such high levels of virus around in their blood, if you're butchering monkeys and you've got any kind of cuts or abrasions on your hands, then this is probably the most likely way that you pick up the virus and it passes to humans. But another interesting thing is that this actually happens very frequently. So at the top of this slide, you can see some sooty mangabees. And this virus has actually passed into humans at least 8 separate times. The virus from chimpanzees has actually passed into humans at least 4 times, and one of these through gorillas. So this is actually a very common event that viruses can actually pass from monkeys into humans. So passing from a virus from an animal into a human is called a zoonotic transmission, and I'm sure you've all heard this more recently with COVID. But generally, when a virus gets into a human from a non-human host, it doesn't replicate very well. And so most of these transmission events only infect one person or maybe a handful of people. And HIV-2 is actually relatively rare. It's mainly confined to West Africa. And it's only infected sort of hundreds of thousands of people. So 98% of the HIV infections around the world have actually come from one transmission event, just from one virus that passed from a chimpanzee into a human. It's now diverged and spread, and we actually now can separate this subtype M, which stands for main, into 9 different clades, and we give those letter names. And these clades are actually geographically distributed. So you'll find different clades in different parts of the world, and this is because they've generally come from sort of one virus being taken to a place and then spreading locally in that area. So where did all of this start? Well, some of the earliest known reports of AIDS, although they didn't call it AIDS at the time, actually came from West Central Africa, which is shown here on the left in this map. And some of the earliest verified samples of HIV actually came from a man who had blood samples taken in 1959 in a town called Kinshasa, which is actually the capital of the Democratic Republic of the Congo. And that's shown in this red dot here on the map. And interestingly, this is about 700 kilometers away from where monkeys or apes, in this case the chimpanzees, that carry the most closely related virus to HIV, this M virus, have been isolated. And these are in southeast Cameroon, which is the red ring on that map. But you can see that these 2 areas are connected by rivers. And in the 20th century, rivers are the major highways, major communication routes. And at this time, this part of Africa in particular went through massive population expansion, and you can see that in the photographs on the right. So Kinshasa actually became one of the largest cities in Africa. And at certain points, there were 4 times as many men in Kinshasa as there were women. They'd come looking for work, mainly. But this made perfect conditions. There were high levels of prostitution, and basically this set the scene for being able to transmit HIV. So using these early samples and comparing it to modern samples and also comparing it to the SIV sequences, scientists have made a kind of family tree of HIV. And using something called molecular clocks, which is where you can sort of see how much a virus or something has diverged from the sort of last common ancestor, then we've worked out that HIV probably jumped species from chimpanzees into humans around the turn of the last century. So HIV is sort of just over 100 years old. And one thing actually that I should also mention, I'll go back a slide, is that the SIV from chimpanzees itself is actually a transmission event from other monkeys. So part of the genes of the SIV in chimpanzee actually come from a virus that infects monomonkeys, which include greater spot-nosed monkeys and moustached monkeys, if you know your monkeys, which I now do. The other half of the genes actually come from a monkey called red-capped mangabeys. And so what probably happened, both of these monkey species are known to be hunted by chimpanzees. So they probably hunted these 2 monkeys, and one chimpanzee ended up eating 2 different monkeys, being infected with 2 different monkey viruses. And then these viruses actually recombined inside the chimpanzee to make a brand new virus, which is SIV chimpanzee. And probably, because chimpanzees are so similar to humans, evolving inside the chimpanzee probably made it easier to transmit this virus to humans. It is a remarkable series of events that have occurred to infect so many people. So from these origins in Cameroon and passing to Kinshasa and that local area, the virus really has spread around the world. And I think this is just a good time to remind you how HIV spreads. So most people contract HIV through sex, but it can also be passed on through sharing needles. And sadly, I'm sure you're all aware from the news that many people contracted HIV through being given contaminated blood products. So I've listed in purple here the bodily fluids that can spread HIV, but these bodily fluids have to come into contact with the mucous membrane or directly into the bloodstream in order to infect. And that's because HIV can only infect white blood cells. So to really understand more about how HIV infects, we're going to learn a bit of the basic biology of HIV. So like all viruses, HIV is an obligate intracellular parasite. So that means that it can only replicate inside a host cell. And that's because viruses are very small and they don't have all the equipment needed to make new viruses. So every type of virus infects a specific kind of cell, and HIV infects white blood cells. So just to give you some idea of the scale of this, if you had a grain of salt, think of a tiny grain of salt, if it was as big as this big blue cube on the slide, then a white blood cell would be the size of that red dot. And if we zoom in on a white blood cell, and that's shown here, then those purple blobs are actually real HIV particles coming out of an infected cell. So they're very, very small. And in fact, they're so small that we can't see them with a normal microscope. So the only way you can see viruses is using an electron microscope. And that can make it tricky to study. So viruses can be as different from each other as animals in a zoo. And so we classify viruses into specific families. And we do this using various characteristics of the virus that I've listed in blue on the right. And the main one there is the kind of nucleic acid that makes up their genome, and then how this nucleic acid is all sort of packaged together to make a new viral particle. And so it turns out that HIV belongs to the family of retroviruses. So what is a retrovirus? Well, they're found in pretty much every vertebrate species that we know of. They mainly cause cancer and immunodeficiencies, and they're very small RNA viruses. They have a lipid envelope that surrounds them. But the interesting thing about retroviruses is they replicate in a pretty unique way. So they make more copies of themselves by using a process known as reverse transcription and integration. So pretty much all living things have a genome made of DNA. So you have DNA, trees, bacteria, everything is DNA. This then gets copied into something called RNA, which is quite similar to DNA. And this is then read and translated into protein, and protein is what makes up organisms. But viruses, uniquely, can also have RNA genomes. And so HIV has its own genome made as RNA. So most viruses then just skip the DNA bit and go RNA protein. But retroviruses have a special enzyme, and this is called reverse transcriptase. And this means they can take their RNA and make a DNA copy of this. And this sort of backward step that goes against the sort of general dogma of how things work is where they get their name retro from. And then the other unique thing they do is they take this DNA that they've made and they insert it into the cell that they infect, into the chromosomes of the cell they infect. So they make a permanent copy of themselves in the cell that they've infected. And we call this integration. And this is carried out by another enzyme of the virus called integrase. But because it sticks its own genes into that cell, that's a permanent infection. You can't get rid of that. So that cell will be infected for the rest of the life of that cell. And this is the main reason why we can't cure HIV. So this is the whole replication cycle of HIV. And I'm not going to go into detail about any of these steps really. But basically, the first thing a virus has to do is get into its target cell. And it does this by binding to receptors, so proteins, sticking out of the surface of the cell. And each virus recognizes specific proteins, and that determines which cell it can get into. So HIV binds to a protein called CD4, and this is why we We call them the CD4 T cells that I had in a previous slide. It also uses a second receptor. In the majority of cases, this is called CCR5, and I will come back to that at the end. But once the virus is bound to the cell that it wants to infect, it releases its core into the cytoplasm of that cell, and then it replicates its genome. And as I just said, this is by reverse transcription and then integration. And then it gets the cell, because it doesn't have all the machinery that's needed to make new proteins and new viruses, so it gets the cell to do that. So the cell makes new viral proteins and new viral genomes, and it assembles them all together to make new virus particles that are released from the surface of the cell in a process we call budding. And this is actually, they push through the membrane of the cell, and they take a bit of that membrane around them to create their own envelope. And this makes what we call an immature particle, and the particle then needs to go through a process that we call maturation to make new infectious particles that can go on and infect new cells. So, like all retroviruses, HIV has 3 main genes, only 3. So, the GAG, POL, and ENV genes. And so the GAG genes code for the structural proteins of the virus that make up the viral particle. The POL gene codes for the enzymes, and I've already mentioned reverse transcriptase and integrase, and there's a third enzyme, protease, which I'll come on to. And ENV codes for the envelope glycoproteins. So these are like the spike proteins of coronaviruses that you might have heard of for COVID. But they basically stick out of the virus, and it's these proteins that then attach to the cell that it's going to infect. And so initially, these GAG and POL genes are make really long, what we call polyproteins. And these long proteins assemble at the membrane at the outside edge of the cell, and they start pushing through the membrane around the cell. And you can see that in panel A in this figure here. Eventually, they push all the way through, and they make this sort of new particle. And if you look closely at panel B, you can see little spikes around the edge of this particle, and those are the envelope glycoproteins sticking out. And then something clever happens, and the protease enzyme of the virus chops up these long proteins into smaller little proteins. And these then rearrange inside the viral particle to create the mature infectious virus. And you can see in panel C that these have a really nice sort of cone-shaped core in their center. And this is the main core of the virus. So this is my diagram of that. And this conical core that you see is made up of this capsid protein, which is shown in green. So each individual protein is highlighted in different color and shape, and they all have important roles in replication in making new copies of the virus. So in addition to these 3 main genes that all retroviruses have, HIV has some extra genes. So it has 2 regulatory genes called TAT and REV, and these are basically help to express the viral proteins, make more of the viral proteins. And then it has 4 so-called accessory proteins or accessory genes, and these are shown in pink and purple here. So accessory proteins aren't actually required to make new virus particles, but they seem to be highly conserved for a start, so they're probably very important. But their role seems to be to overcome basically the cell's natural defenses, so proteins in the cell that would otherwise inhibit replication of the virus. And so these accessory proteins go around and find these proteins that would block their replication and get rid of them. They basically usually destroy them. And interestingly, HIV-1 and HIV-2 actually have slightly different accessory proteins. And this may explain why HIV-2 is slightly less pathogenic than HIV-1. And it also may explain some of the properties of these accessory genes, may explain why some viruses are transmitted more easily than others. So they're quite interesting to study, and actually my lab are interested in understanding what these accessory proteins do and how they work, and the ones that we're currently focused on are highlighted here. So as well as that, we're also interested in the stage of genome replication, kind of shown here. And this is because, as I said, this integration step is really key as part of the replication process of HIV, but this is the reason that we can't cure HIV. So we're really interested in all the processes that lead up to this point of integration. And I've told you that the virus is very small, and it doesn't have it has all the information needed to make a new virus in its genes, but it doesn't have all the equipment it needs. So to do that, it needs to use the cell's own machinery. And this is a busy slide. This just shows you some of the proteins and pathways that the virus actually uses to undertake all this replication. And actually, you can see at the bottom there, this slide is actually 15 years old. So we now know a lot more proteins and pathways that HIV uses. So just quickly to say how we study this, actually, we use lots of different techniques, which are highlighted in green here. So from virology, molecular biology, cell biology, biochemistry, right up to some protein structure. And so to help us do that at the Crick, we have lots of science technology platforms, STPs, that are experts in certain techniques, and they're sort of shown in purple here. And so we interact with a lot of people at the Crick to help us with our work to understand what's going on. And one of the proteins that I've already mentioned that we're most interested in is this capsid protein. And so about 1,500 molecules of proteins of capsid actually arrange themselves together and assemble into this quite beautiful lattice that makes up the outside of the core of the virus. And in the bottom here, we've got an electron micrograph of individual cores of HIV. And it's this capsid protein that seems to bind to a lot of these cellular proteins and be involved in working in a lot of these pathways to help replicate the virus. And because it's so essential for viral replication, this makes it a really good drug target. And in fact, the latest drugs for HIV that were only actually approved at Christmas target this capsid protein. So that brings me nicely onto different treatments for HIV. So once it was first discovered that HIV was a retrovirus, as I've said, this unique process of replication meant that the first targets for drugs that people came up with were against this reverse transcriptase enzyme. So the first drugs against HIV were given to people in 1987. The first drug was called AZT. But within a couple of years, it was reported that the virus had become resistant to AZT. And as I told you, you can make billions of virus particles every day, and this means that the virus has lots of opportunity to kind of evolve and adapt and change, and so it can get round drugs very easily. But until 1993, this was the only drug that we had against HIV. And what that meant is that people kept dying. And so this graph shows you the number of deaths from different causes from young people in America, and the red line is HIV. And so you can see that by 1994, HIV was the leading cause of death in 25 to 44-year-olds in America. So really something had to happen. And activism actually played quite a large part of this. People got out and protested and demanded new drugs to stop HIV. And actually the scientific community and the big pharma companies responded. And so they started to develop new drugs. And the first new drug to be developed was against this protease enzyme that I mentioned is responsible for cutting up these polyproteins and making the immature virus into an infectious virus. They also came up with new drugs that targeted reverse transcriptase, but in different ways. The virus would have to adapt differently to overcome those. But the biggest breakthrough came when actually people were given these drugs in combination. And this we call highly active antiretroviral therapy, or HART. And you can see what they're stood in these graphs here. So in the first graph, the green triangles, you can see that if you just give people AZT, then the virus levels drop for a few weeks, but then they start to go back up again as the virus finds ways to get round these drugs and replicate normally. So then in 1994, people started, when we had a new drug, basically, people were given two drugs. This had a bigger effect on the virus. The virus levels dropped more, but they still carried on going back up. So the breakthrough in 1996 was to give people three drugs. And when the virus was up against three drugs, it just couldn't adapt to get round all three of them at the same time. And for the first time, and this is 15 years after AIDS was first reported, for the first time we could now stop people from dying. So this was a major breakthrough. And what it meant was that the virus levels remained low, and this gave the body time for the immune system to recover a bit. And so that's really what was behind all this. But of course, the drugs only work if you can afford them. And so while we could stop people dying in the West, in sub-Saharan Africa, which had the major burden of HIV, where there was the most people living with HIV, these drugs were too expensive. And so millions of people kept dying. And so it was around the year 2000 that somebody said, well, this isn't right. And a major player here was Dr. Yusuf Hamid, who ran a generic drug company in India. And he basically turned around and said, well, I can make these drugs much cheaper than the pharmaceutical companies. And so he did. And actually, the pharmaceutical companies let him do that. They didn't sue him. And then importantly, the United Nations set up a global fund to pay for this, because even these generic drugs were kind of too expensive for people in Africa to afford. So the United Nations set up the global fund to treat not only AIDS, but also tuberculosis and malaria, which are the other big killers, and in combination with HIV, were causing massive problems. And then whatever your politics, actually President Bush did something quite amazing. And he set up PEPFAR, which is the President's Emergency Plan for AIDS Relief. And he put a huge amount of money in. And so this meant that actually people in Africa for the first time could get hold of these drugs. And you can see what this did for life expectancy. So from the early 1980s, life expectancy was dropping rapidly in many of these African countries. But then once you could get therapy to them, then life expectancy started going back up again. And now we have over 25 different drugs against HIV. So as well as these drugs, we've got new drugs that target this entry process. We've got some integrase inhibitors. So this is the integrase enzyme of the virus. And as I said, the latest drugs target this capsid protein. And so we've also managed to improve treatments with time. So initially, people used to have to take handfuls of drugs several times a day. But now we've got it down to 1 tablet once a day. We've also managed to tweak the drug doses, and with the new drugs as well, they're less toxic. And so altogether, what we've been able to do is to reduce people's viral loads, so the amount of virus that's circulating around in their bloodstream, to undetectable levels. So if you take your drugs properly, then we've got the virus down so low now that we can't detect it. And what this means is that they can't pass it on. So the big message is that undetectable equals untransmissible, U equals U. And so potentially now we have the tools to stop new HIV transmissions. And we can also use these drugs in another way, so we can actually use them to prevent infection. And so this is called PrEP, or pre-exposure prophylaxis. And people take the drugs to protect themselves. And the UK was actually one of the countries that did one of the landmark studies in this, to show that there was an 86% reduction in people catching HIV if they were taking these drugs as a preventative measure. So we can actually now treat everybody, theoretically, with HIV. And the take-home message is that now HIV is no longer a death sentence. We can actually treat people. They can live very healthy lives, and their life expectancy is pretty much the same as the general population. So what do we still need to do then for HIV? Well, as I said, the drugs only work if you can take them. But in order to take the drugs, people really need to know that they've got HIV. And the problem is that there's this long asymptomatic stage where people don't really feel sick and they don't know that they've got the virus. So there is a big drive to get people tested. And this is just Prince Harry showing that it's fine to get tested live on TV. But this is working. So actually, the number of people living with undiagnosed HIV in the UK is actually falling. And we are on target to reduce the number of new HIV transmissions by 80% next year. and hopefully by 2030 to stop all new HIV transmission. And a major point of this is actually that we've introduced opt-out testing. So this means that when you go into hospital, you can now be tested for HIV and you have to opt out if you don't want that. And that means that we can diagnose people who didn't realize they were HIV positive and get them onto drugs quickly, stop them from spreading it to anybody else, and also treat them to stop any complications. So treating people with drugs is working to stop transmissions, but a better way, that's still quite expensive, giving people drugs all the time for that. So a better way would actually be to have a vaccine for HIV. And after the COVID vaccine success, where it only took 11 months from identifying the virus to coming up with a vaccine, people always ask, why after 40 years do we still not have a vaccine for HIV? Well, that's because it's very complicated. It's a lot more complicated than COVID. And it's complicated for a variety of reasons, but one of them is that there's no natural immune response to copy. So vaccines normally work by giving people a little bit of the virus, a single protein up to sort of a weakened version of the whole virus. And this sort of kickstarts your immune system into reacting against it, finding a way around it, so that when you actually see the proper virus, then the immune system just suppresses it straight away. But this isn't what normally happens in an HIV-infected person. No, they do mount an immune response, but it doesn't clear the virus. And as I already said, monkeys have very high levels of virus in their blood. They don't seem to control it at all. So there isn't really a natural response to mimic, and we don't really know sort of what to tell the immune system to try and overcome it. So that's one problem. The other problem is that HIV is highly variable. So we've talked about how the virus can easily evolve to overcome immune defenses or drugs. But there's actually as much variation in a single person of different HIVs at any one time as there is variation of something like the virus that causes COVID in the whole world. So we're up against a much bigger problem, and the vaccine would need to work against all of those different variants to work properly. And then, of course, we have the problem that HIV actually infects the immune system. So it infects the very cells that are supposed to be defending against the virus. But the other thing that I've mentioned a few times now is this integration step. So basically, you have to get every single virus, because just one virus will give you a permanent infection that we can't get rid of. So this is sort of similar to cancer, where you have to clear all of the cancer cells to make sure that the cancer doesn't come back. And you can see this here in this graph, that we can give people drugs and we can suppress the virus levels to undetectable, but the people have to take those drugs every day. Because if you stop taking the drugs, then the virus rebounds and viruses get back up to the levels that they were before. So why should we cure HIV? Well, first of all, there's the problem of drug resistance, which I've mentioned, that the virus can get around the drugs. And so far, we've been able to stay one step ahead of that. We've come up with new drugs. But eventually, this may cause even more of a problem. Secondly, we're sort of victims of our own success, really, because as therapy gets more successful, people get more reluctant to take a tablet for something that when they don't really feel ill. So having to take a tablet for something every day, you end up with sort of adherence issues. And there's further issues where if people can't get hold of their drugs, this is an issue. And in certain places, this can be a real problem. So for example, when Ebola hit Africa and there was the Ebola crisis, the HIV clinics were shut and people couldn't get their medicines. So there's lots of social economic reasons as well. Also, people are living longer, which is great. But this means that they're on drugs for longer, and we don't really know how that affects them, particularly as they get older and they start taking medication for other ailments. We don't know how the drugs interact. Obviously, it's very expensive. The more and more people that are living with HIV and they have to take drugs for the rest of their lives, that's quite a big economic burden. And then finally, there is still a stigma attached with being HIV positive, and that's actually something we can all do something about. Because really, we shouldn't be letting stigma stop people accessing medicine and saving lives. So I guess the big question is, can we cure HIV? And I've talked about this integration step. But yes, we have actually managed to cure a few people. So the first person cured of HIV was called Timothy Ray Brown. He's here on the left. This was in 2009, this was reported. So Timothy was HIV positive and then diagnosed with leukemia. And for his leukemia, he required a bone marrow transplant. And so when you do a bone marrow transplant, you basically kill all of the white blood cells in a person and then you replace them with white blood cells from somebody else. So they killed all of his white blood cells, including all the cells that contained HIV. But the really clever thing is they gave him some cells from a donor who had a specific mutation called CCR5 delta 32. So if you remember, I said that CCR5 is actually one of the proteins on cells that the virus binds to get into cells. And people with this delta 32 mutation don't make any of this protein. They seem to be perfectly healthy, but it means that HIV can't get into their white blood cells. So they managed to find a donor that was a bone marrow match that also had this mutation. And so they gave back cells that couldn't be infected with HIV. So if there was still any HIV in his body, it didn't have any new cells to infect. And it worked in this person. Unfortunately, they tried it in other people and it didn't work. But the other thing is that this CCR5 delta 32 mutation is really rare. It's found in about 1% of Caucasians, so people of European ancestry. It's even rarer in other people. And so it took another 10 years before that they could repeat this in another person, called the London patient because he was treated here. And he had a different cancer and he had slightly different treatment, but we managed to cure him as well. And so this has given us hope that it is possible that you can actually cure HIV if you can remove all of the cells that are infected with HIV and then possibly replace them with cells that can't be infected. So bone marrow transplants have their own risks and there's a high mortality rate. So this is not a general treatment. But could we apply what we've learned here to being able to come up with a general treatment? Well, there's something called kick and kill. And the idea here is that we could try and identify all the infected cells and specifically just kill those. That actually is a bit tricky because in general, the infected cells, particularly if they're not actually churning out virus, if the virus has sort of gone dormant in these cells don't look any different to the uninfected cells. So trying to identify which ones are infected is quite tricky. We also don't know where they hang out because they're probably in lymph nodes or other parts of the body that aren't really easily accessible. So far this hasn't really worked. So as an alternative, we could think about something that's not a complete cure, but maybe something that's a functional cure. So in this case, people would still have HIV genes in their chromosomes, but they wouldn't be able to make new viruses. And so sometimes this is called block and lock. So we block the virus from being able to come out and make new viruses. We kind of lock it into this dormant state. But again, we're trying, but this hasn't really been successful yet. So there's obviously still work to be done to finally actually cure people of HIV. There's still a lot to learn about the virus. But I just wanted to leave you with a positive message because actually we've actually learned lots from HIV and scientists have now started to use HIV as a way to treat other diseases. And so what we can do is we can take out the harmful genes, the viral genes from the virus, and we can put in helpful genes that we want instead. And we can also change the envelope proteins on the surface of the virus so we can infect different cells, not just white blood cells. And so what we can do is we can turn all the things that I've been talking about that are really bad, I mean, we can't treat the virus and we can't cure the virus, we can turn them to our advantage and we can use them. So we can put helpful genes into virus particles that we call HIV vectors, and we can use these to deliver new genes into cells that we want in the form of gene therapy. And so people have actually now been treated with HIV virus particles carrying genes that replace faulty genes in their own bodies to cure them of other diseases. And we can use the same kind of vector system in CAR T-cell therapy, which you may have heard of, which is where we basically train a person's immune cells, their white blood cells, to go and kill cancer cells. And we can use these HIV vectors in order to manipulate these T cells and modify them to become CAR T cells. So although HIV is a devastating disease, we've actually been able to also find some benefits of it that we can now use to help other people. So that was a crash course in HIV. So thank you very much.